Hello, everyone. I'm Jason Prahl with the Human Longevity Project, and I want to welcome you to this mind blowing webinar on the immune system with my good friend Kiran Krishnan. If you're not familiar with Kiran, he is a research microbiologist and has been involved in the dietary supplement and nutrition market for the past 16 years. He comes from a strict research background, having spent several years with hands-on R&D in the fields of molecular medicine and microbiology. And as part of his formal education, he actually did some postgraduate research and advanced coursework in molecular biology and virology. So this is kind of what makes Kiran such an expert on this topic of boosting your immune system naturally. It is this expertise that prompted him and his research team to formulate a unique trio of products that have a tremendous synergistic effect that offer you added protection beyond some of the more traditional herbs and supplements on the market. And for a limited time, you can actually take advantage of a discount we're offering on these products from Microbiome Labs. So make sure to stick around for that, as well as the great Q&A at the end. So with that, here's Kiran Krishnan. Enjoy.
So thank you, everybody, for coming on, and thank you, Jason, for this opportunity to talk about this really important topic. When I was talking about this with Jason beforehand, I was like, I'm going to put together some slides. I was thinking I'll put like 6 or 7 slides together just to convey the most important components of the message. And then I start working on this, and I'm like, well, that's really important. Well, that's really important. That's really important. Now I have something like 39 slides. So I promise I'll get through it all. But the most important thing is I want to make sure that everybody really understands this information, because in many cases it's absolutely critical to how you will fare when your immune system is challenged with something. And as Jason mentioned, the whole message here is the immune system and the microbiota are essentially one and the same, right? We think of them as completely different systems. And when you see parts of this presentation, what you'll come to realize is they essentially function as one and the same in a sequence of events to protect the human host. They are constantly in communication, and you'll see why. And then most importantly, towards the end, I'll give you 2 really main steps that you can do, things to focus on that will be critically important to strengthening your immune system naturally. And of course, many of you already know about some of the key micronutrients like vitamin C and zinc and magnesium, and those things are all perfectly good and important for your immune system. But this is going to give you more depth, and this is going to give you a deeper understanding of the core things that are holding back your immune response. Right, the dysfunctions we're going to talk about within the system that compromises immune response cannot be overcome by just supplementing with vitamin C. Right, so that's the critical thing I want people to understand from this talk. So let's jump into it. The first and foremost, really, to understand this entire conversation, you have to keep realizing and reframing your mind that the human is not a human as we used to think of it, but we're more a superorganism or a holobiont. A holobiont is a superorganism. So we are a construct that's made up of trillions upon trillions of microorganisms, right? We have over 40, 50 trillion microorganisms in our system. And that's far more than human cells within our system itself. And even more importantly, we have a lot more microbial genetic elements within our system than we have human genetic elements. So about 90% or more of all of the genetic information in our body comes from microbes and not our own human cells. And the vast majority of metabolic outputs come from the microbiome, not from our human code itself. So we are a walking, talking rainforest, and a compromise to our ecosystem, our inner ecosystem, is what drives disease. Right. And of course, our immune system is a very intimate part of this ecosystem. This is all derived from a process called symbiogenesis, which is really the result of a permanent coexistence of different bionts, so different organisms, including the human Homo sapien, which then come together because of necessity to form a holobiont. And so this is the interaction between the host and the microbiota. I'll get a little bit more into how the microbiota and the host immune system interact more specifically But this is a general concept you have to understand in order to understand how the immune system works. Now, looking at the kinetics of the immune response, right, there's really three major phases to the immune response. The first one is this here, which is the innate immune, which is the early response to an incoming pathogen that can kick off and happen within hours of a new virus or new bacteria coming into your system, or an old virus or old bacteria, one that you've seen before. The innate immune system responds first. Now, in the late immune phase, there are these other cells called macrophages and dendritic cells that are antigen presenting cells. They end up showing up, and then they facilitate the immune response towards what we call the adaptive immune response. Now, this takes days, right? But in the adaptive immune response, what happens is we produce B cells that convert into plasma cells. These plasma cells now make antibodies against whatever the offending organism was, right? So now your body has these drones floating around all the time, ready to neutralize that invading pathogen the next time it comes in. So the second time you see it, it can take care of it much faster. Now, you also have memory T cells and cytotoxic T cells that can also have some long term memory on these invading pathogens that can go and take care of it the next time you see it again. But this is the kinetics of the immune response. This first part occurs within hours. And then within the first day or two, you start getting the second part, this intermediate part, and then we move into the adaptive immune response, which in a normal, healthy individual might be day 3 or day 4. Right now, a couple of critical things. Inflammation is a critical component of the innate immune response, and I'll show you why later on. However, in order to move from the innate into the adaptive, we actually have to have a step of anti inflammation. We have to de escalate and bring down the inflammatory response in order to move to the next phase, which is the adaptive immunity. Now, one of the things to point out is that the innate immune response is non-specific. These immune cells know how to get to an area where something is wrong, but it's hard for them to know exactly what is wrong, right? So think about these guys as bug killers. Let's say you've got a massive mansion of a house, and there are thousands of windows all over that mansion of your house, and all of those windows are open, right? And then you've got bugs that might fly into the window, and you've got a whole army of bug killers that can respond to the presence of a bug entering in the window. The innate immunity would be like the fastest people in that anti-bug army that can run up the stairs the fastest, that pick up the earliest signals, and they get to that window where the bug is coming in first. The only problem with them is their tool that they have to kill the bug that's coming in is a blowtorch, right? So they're going to be pretty effective in blowtorching and killing some of the bugs coming in, but they're also going to burn the wall and burn the window a little bit, right? So if they are allowed to function for too long of a time, you're going to burn the whole house down. That's one of the ways in which the immune system goes haywire. In fact, in the pandemic, that's one of the big issues, is something called a cytokine storm. That is the innate immune system going crazy, blowtorching the crap out of the system, and not moving into this anti-inflammatory and adaptive immune response. Because these intermediate people here would be a little bit slower than the innate people. They would show up, they would come, they do have some tools of their own that are not blowtorch like tools, but the main thing that they do is they take pictures of the bug, and then they come back to these cells, the B cells and T cells, and tell these B cells and T cells exactly which bugs have come into the window. And then based on the pictures that they tell, or the presentation that they give, the right B cell that has the specific tool for just that one bug, or the right T cell that has a specific tool for just that one bug, gets recruited to that window. And so that way, there's a specificity to the response. We no longer have to blowtorch the whole area to kill that one bug. These guys will have bug killer spray that's only specific to that one type of bug. So it doesn't damage anything else, and it's highly specific. Hopefully that analogy help make sense of this whole immune kinetics. And I know it sounds a little elementary, but that's, if you understand that, then you kind of understand how the immune system works, right? So these antigen presenting cells are critical, the innate immune cells are critical, adaptive immunity is critical, but you can't have any one of these going on for too long. And you cannot have a stop in this process. And in order for the process to proceed, you have to have inflammation, then you have to have anti-inflammation to move on. Now, there are three other components to remember while we're looking at immune kinetics. The first one is surveillance, right? These cells, the basophils, mast cells, natural killer cells, neutrophils, even macrophages and dendritic cells, are all part of the surveillance team of your immune system. These are the cells that are circulating around your body all the time, looking for invading pathogens. And I'll talk about how they find and identify invading pathogens. But that surveillance, immune surveillance, is critically important to maintaining a healthy immune system. It's very easy for your immune system to become stagnant, to turn down its activity when the host doesn't need, when it presumes the host doesn't need protection, right? So we don't want the immune system to always be in the sleep mode. We want the immune system to have a certain degree of functional readiness. It doesn't mean the immune system should be raring all the time, because that overactive immunity is a problem as well. But we want it to have a certain degree of functional readiness, and that is the surveillance capability of the immune system. Then the second part that's really important is this anti-inflammatory part, and that's called the de-escalation, right? Because these guys are going to go crazy with the blowtorching and escalate the immune response. Then we need a de-escalation of that immune response so we can move to the more specific adaptive immune system. Now, in the blowtorching phase, one of the big issues that occurs is there's a lot of tissue damage, right? So when you have that kind of tissue damage, you also need a cleanup crew to come through and start basically cleaning up our own dead cells, parts of microbes, parts of viruses, basically the catastrophe that's left after this really violent part of this initial immune response. Like the blowtorching, you're going to have ash everywhere. You're going to have parts of the wall that are burned. You're going to have parts of the wall that are falling on the ground. You need to clean all those things up as well. There's a few reasons for that. Number one, because if we don't clean up, especially our own cells, components of our own cells, the immune system can mistakenly start initiating an immune response against components of our own cells. And that's one of the ways that one could develop an autoimmune response after an infection. That's not an uncommon thing, right? And in fact, with COVID, one of the big issues with COVID and why we have these COVID long haulers is because COVID is one of those types of infections that can initiate an autoimmune response on your own tissue because of all the damage that's created through the robust innate immune response against the virus, right? So the cleanup crew, the rebuilding is absolutely critical as well. So remember these three things: the surveillance component, the de-escalation, and the cleanup, because that'll become important later on when we talk about what are the things that we can do to improve our immune response. Now, where is all of this immune battle going on in the body? Well, it's in the mucosa. The mucosa is the largest surface area in the body, right? It covers every bit of the inside part of your body. It's about 400 square meters. For those of you who are not familiar with the metric system, that's about 4,000 square feet, right? Compare that to only about 2 square meters of the skin. We used to talk about the skin as the largest barrier or the largest organ in the body, but it's not. The mucosa on the inside is a couple hundred times larger than our skin in terms of surface area. So imagine if you had a 4,000 square foot apartment or house, you'd be very happy about it. That's a lot of surface area. All of that is folded into your system called the mucosa. And anytime anything enters your body, whether it's through your skin, your digestive tract, your eyes, your ears, your nose, your urogenital tract, it's always going to enter into the mucosa, right? So there's a mucosa in every part of your body. And so most of the microbes that live in your body live in the mucosa, and most invading pathogens that are going to enter your body enter into the mucosa. So the immunobiology of the mucosa is absolutely critical. And this is, again, the largest surface area in the body. It's also the largest portion of the immune system. Most of the mucosa is actually found in the gut. So the entire lining of your 28 feet or so of gut is actually covered in mucosa. And if you were to actually unravel the entire surface area of just your gut, it's the size of a professional tennis court, right? So you know how big that can be. So that all of that is just in the gut part of it as well. And as I mentioned, it lines the entry into every part of the body: respiratory, digestive, reproductive, skin, and so on. It's the largest site of immune sampling because, again, everything that enters the body, whether it's food, chemicals, viruses, bacteria, and so on, have to enter through the mucosa. And since your microbiome is in the mucosa, your microbiome is checking things out first that come into the system. And of course, most of your immune tissue is in your mucosa as well. So your immune system is also monitoring everything that enters into the body through the mucosa. And of course, the mucosa is covered in microbes because your microbiome lives there, right? So that is the part that's really interesting when you think about it. The mucosa is where all of the pathogens enter. That's where an infectious agent is going to enter into your body. It's entering into a huge surface area, 4,000 square feet, folded up inside your body. Every square millimeter of that 4,000 square feet is already covered in microbes, in bacteria, viruses, fungi, and so on. So how is it that the surveillance part of the immune system is supposed to manage this system? It's so complicated. If you look at this as an analogy, right? So let's say this is your mucosa, and these are all your microbes standing shoulder to shoulder, high density, no open space at all. That amounts to over 40 trillion microbial cells in your mucosa. When you look at the 40 trillion or so microbial cells in your mucosa and you compare that to about 200 million immune cells that you have to survey the mucosa, that is a 200,000 to 1 ratio of immune cells to microbial cells that already exist in the mucosa. So for every one immune cell, its job is to maintain surveillance on over 200,000 microbial cells, right? So how can that one immune cell in that sea of 200,000 ever dictate a virus or bacteria, or sorry, detect a virus or a bacteria or something that's actually going to be harmful versus the natural viruses and bacteria and all that live in the system, right? So imagine you've got a crowded system like this, and you've got here now, among the sea of normal microbes, you've got one here that's egregious. You might have another one here that's egregious, another one here that's egregious. How would the single immune cell that's surveying 200,000 plus microbial cells pick out this one out of that sea? The only way it can do that, and this has been a massive confusion in the world of immunology for years, right? Like, how is this done? How does our immune system that lives within a sea of microbes figure out which microbes are friend and which microbes are foe? The only way it can absolutely do that is if it has a neighborhood watch system. And the neighborhood watch are the other microbes, the commensal microbes that live in this system. They are the ones that are the eyes and ears of the immune system. It's absolutely impossible for a single immune cell to monitor 200,000 or so microbes every second of every day. The only way they can do that is if the majority of those 200,000 microbes are on the side of the immune system. And they are the ones that would alert the immune system if something enters into that space that shouldn't be there, right? So they are acting as a neighborhood watch. That's the really important part to understand about the relationship between the microbiota and the immune system. Is the microbiota as the eyes and ears of the immune system. It's the first thing that detects a change in the mucosa. This is an illustration of just some of the crosstalk that occurs between the immune system and the microbiota. Now, this is the kind of stuff, and I promise you, I'm not going to go through this whole chart because I'm sure it's going to give somebody a migraine. But the whole idea here is the discovery of the microbiome immune crosstalk answered so many fundamental questions about how do we exist? How do we coexist with microbes in our system when our immune system is designed to fight against microbes? Right. So this kind of crosstalk across from the mucosa up here into the circulatory system, and all of this information sharing between the microbiome and the immune cells are what allow us to have an open conversation. Our immune cells have an open conversation with the microbes that exist there naturally, and how those microbes can support the immune system to try to educate and train it on how to respond and when to respond. Right. So let me give you a few examples of that very idea. So take a norovirus infection, for example. Right, there are lactobacilli and other commensal organisms in your system that trigger the release of interferon beta and interferon gamma, which then alerts the innate immune system to the presence of the virus. Right. So the moment the virus enters a system, the first thing that detects the presence of the virus is the microbiota, and the microbiota sends signals or flares to your immune system in the form of interferon beta and interferon gamma to alert the immune system that, hey, something is going on here. I need you to come and pay attention to it. So it's acting as a way of honing the immune system into the area that is getting attacked. And then vitamin A, for example, in this case, provides a substrate for the commensal bacteria to make these interferons. So one of the ways that some of these micronutrients help us is they provide building blocks or energetics or components of the microbiome to actually utilize to signal to the immune system and so on. Here's another example on a rotavirus infection, bacterial flagellin. So there's a lot of bacteria in your microbiota or even in other parts of the body that naturally have flagellin. And so these commensal bacteria's flagellin, they activate the expression of something called pattern recognition receptors, PRRs. The activation of PRRs triggers the activation of something called toll-like receptor 5, so TLR5, which then can stimulate the release of interleukin 22 and interleukin 18. And it stimulates the release of these two interleukins from your immune cells. So a component of one of your commensal bacteria can cause your immune cells to release interleukin 22 and interleukin 18. And look at what these two interleukins do. Interleukin 22 helps repair the damaged epithelium. So remember, a rotavirus infection occurs in the gut, right? So it's a gut infection. People will vomit, have lots of diarrhea, typically affects kids a little bit more than adults, but it's not a pleasant condition. You're going to be in bad shape for a few days because you're going to be dehydrated. You're not going to be able to hold anything down. You can have really high fevers and so on. You can be vomiting all the time. But your gut lining is getting infected. The epithelial cells in your lining are getting infected by rotavirus. And the first thing that sees the rotavirus come in and start infecting the lining of your gut is your bacterial flagellin, which then stimulates your immune cells in the gut lining to produce these interleukins. And these interleukins, in the case of interleukin 18, it induces apoptosis in infected cells. Remember, a viral infection, the virus comes in, takes over your cellular machinery and causes your cell to produce more virus. So one of the ways that your body defends against losing more and more of its cells to becoming a viral factory is those cells are induced to undergo apoptosis, meaning destroy themselves. So they stop becoming a viral factory, right? So the bacteria in your microbiota are inducing this apoptosis in infected cells. And then with the release of interleukin 22, it helps repair the damaged epithelium. Because once one of your intestinal cells gets damaged and induces apoptosis, you're going to have to replace it with a healthy cell, because if you don't replace it, then you're going to have a gap there and you're going to have severe leaky gut. And so the induction of replacing the cell also comes from your commensal bacteria. Bifidobacteria breve and in combination with things like galacto-oligosaccharides and fructo-oligosaccharides have been shown to prevent rotavirus infection at all, the rotavirus we just talked about. And it does it by increasing interferon gamma, IL-4, TNF alpha, toll-like receptor 2, all of which increases the mucosal immune defense system. So if you are a person that got exposed to rotavirus, but you had really healthy, strong microbiota, and that microbiota was producing things like short-chain fatty acids and all that, you would be more protected against the infection from that virus than somebody that doesn't have an equivalently strong gut. And it does it through these factors, right? So that's really important. Commensal bacteria also produce things like short chain fatty acids, and those are required to increase the production of the mucous membrane. So the mucosa has to keep regenerating itself. Remember, that's the largest surface area in your body. All of your microbiota basically lives in the mucosa. There's, of course, microbiota in your cerebral spinal fluid, in your blood, and so on. But the vast majority of it lives in your mucosa. And that barrier that the mucosa provides is a critically important barrier. And so commensal bacteria are the ones that stimulate the release of butyrate that increases the production of that barrier, because that barrier gets damaged over time. In addition, commensal bacteria can also increase the synthesis of antiviral compounds like reactive oxygen species and defensins, which then prevent local viral infection, right? And replication as well once an infection has been created or established. So again, your commensal bacteria support and defend you by stimulating your immune system to do certain functions. And some of these are very critical. And it's not just in the gut, right? So when you look at influenza infection that affects the lungs, commensal bacteria trigger the release of inflammasomes. Inflammasomes are a type of component within your immune system, which is a potent defense mechanism against influenza replication. So these inflammasomes will actually induce those dendritic cells. Remember dendritic cells from that kinetics chart? Those are the intermediate cells that come in the late innate phase, and then they're the ones that make a copy of whatever is infecting the host and then take it to the T cells and B cells. So dendritic cells are critical to eliciting a normal immune response. And so the inflammasomes that the microbiota, the commensal bacteria, induce the release of will cause dendritic cells to migrate to the local lymph nodes, where they will then stimulate and activate influenza-specific T cell response in the lungs. Right, so shuttling the response to the more potent and less damaging early adaptive response. So what does this mean? This is really, really fascinating. As it turns out, if you get exposed to the influenza virus in your lungs, somehow the lung microbiota picks up the presence of the influenza virus. It translates a signal to the gut microbiota, and then the gut microbiota induces the release of inflammasomes and recruitment of the dendritic cell to the site of action. So think about that communication. Virus comes into the lungs, the lung bacteria picks up the presence of the virus. Somehow the lung bacteria communicate to the gut bacteria. That part we don't even know yet how they do that. And then the gut bacteria recruits immune cells to go to the lungs. I mean, it's absolutely insane. It's a central command center for everything that seems to be occurring in the body. Here are some other examples. The gut microbiota also regulates the respiratory mucosa. So the gut microbes regulates what the mucosal tissue looks like in the respiratory tract. And this is especially important in response to influenza infection. And it stimulates the release of things like more IgA, which are the antibodies that are in secretory fluids that can help protect against infections. It activates Th1 and cytotoxic T cells to deal with the infection itself. And when influenza virus is present in the lungs, the gut commensal bacteria increase the presence of innate immune cells, which I mentioned earlier. And they do this by sending out a bunch of flares like interleukin 33, interleukin 1 alpha, interleukin beta, interferon gamma, and so on. So this causes more natural killer and dendritic cells and macrophages to end up migrating up into the lungs. And when virus is low or not present, right? So this is what I talked about is what occurs when the virus is present in your lungs. But when the virus is not present or it's low at low levels, gut commensals do the exact opposite by releasing anti-inflammatory compounds like IL-10. Remember, one of the ways that the gut microbiota helps you with the influenza infection is by releasing inflammasomes, which are pro-inflammatory. But while there's no infection, the gut microbiota releases anti-inflammatory compounds that also make its way to your respiratory tract. This balancing act is a clear example of this whole gut-lung axis, right, where microbes in the lung communicate with microbes in the gut and inform the presence of a pathogen. That's an amazing chain of command that occurs inside your body, right? We have outsourced critical defensive measures to microbes that exist within us. I mean, just think about that, right? Our immune system is arguably one of the most important systems in our body because without the immune system functioning, we would die from simple infections pretty quickly and early on in life. And as important as the immune system is, we've outsourced so much of its capability to microbes that live in our system. Here's another example. Staph aureus on airway surface recruits monocytes. Those are another one of those antigen presenting type cells that mature into macrophages. So remember in that chart, you have macrophages and dendritic cells that are the antigen presenting cells. So monocytes are the immature form of the macrophage, and it does it through the activation of toll-like receptor 2 during lung infections. This leads to reducing and damage of the lung tissue in acute infections, because if the lung tissue gets blowtorch, then you're going to be in a heap of trouble even once you clear the infection. That's one of the things that's occurring in COVID, where you get thickening of the basement membrane and fibrosis in the lung. That is the scarring of the lungs from the infection, and it, you get your acute part of the infection is really robust. So reducing the acute part of the infection when your lungs get infected is a real big benefit. And as it turns out, Staph aureus is one of the bacteria that stimulates that. Respiratory commensal bacteria like Corneum bacterium can modulate things like toll-like receptor 3's antiviral response to RSV by enhancing the production of TNF alpha, interleukin 6, interferon gamma, interferon beta, and so on, and also through increasing T cell proliferation. So, you've got microbes that stimulate your immune system to respond. And then finally, things like butyrate that are being produced in the colon from commensal bacteria can lower inflammatory damage post early innate. That means at the end of the early innate, as you're moving into the adaptive, by activating things like GPR, G protein coupled receptors on cell surfaces, which then stimulates the release of interleukin 22. And remember, interleukin 22 is a signal to repair the system, right? So butyrate, which is a very important compound made predominantly by bacteria, keystone bacteria in your gut, bacteria like Faecalibacteria prausnitzii, certain types of bifidobacteria. Remember those names because those will become important towards the end. But those microbes are making things like butyrate, which then will actually allow for the repair and regeneration of new tissue and reducing the inflammation. So, here's another example, Lactobacillus crispacus, which is a dominant lactobacilli in a healthy vaginal canal in women. And when it is dominant, it can actually decrease HIV infections. This was a study done in South African women where they showed that certain women had a much lower propensity to pick up HIV infection even with unprotected sex. And they found that these women tended to have higher levels or dominance of Lactobacillus crispacus in their vaginal canals. And so, of course, the bacteria is somehow protecting against the virus's ability to infect. Now, when you don't have a proper functioning microbiome, your immune system cannot respond. It's not that your immune system is compromised in some way. In this extreme case, if you have no microbiome, your immune system will actually cease to respond. And in fact, this study here that was published in May of 2020 is a follow-up to a number of studies that were being done about 10 years prior, 8 years prior. And what they've basically found is that the microbes in your microbiota, right, they calibrate the activation threshold of your innate immune response. What does that mean? Well, they're the ones that set the threshold level that the innate immune cells, so your natural killer cells, your dendritic cells, macrophages, all of those cells, it sets the threshold for which the innate immune system responds to. If the presence of a pathogen or inflammation was below that threshold, they don't respond. If it goes above that threshold, your immune system does respond. And setting of that threshold is based on your microbiota. In fact, the 2020 paper looked at something called no-biotic mice, which are mice that are born without an immune system, without a microbiome. They have their full immune system. They have their dendritic cells and macrophages and all that. And when they introduce a viral pathogen into these mice that don't have a microbiota, the dendritic cells and macrophages and all just sit by and watch the cells get infected and watch the cells burst open, and more virus comes out, and then more virus goes through and infects other cells, and the immune system does not respond. Why does it not respond? Because there's no microbiome. The immune system is sitting there waiting for signals from the microbiome in order to go and respond to that viral infection. And sure enough, in the case of that study, in those mice, when they implant a microbiome back into the mouse, then the mouse's immune system responds immediately to the presence of a pathogen, right? So it's really mind-boggling that our immune system would cease to respond, even though a pathogen is present, damaging our cells, if it doesn't get the right signal, in this case, type 1 interferon from our microbiome, telling it that it has to respond, right? So when you have your immune system, when you have your microbiota and it's functioning properly, you've got high diversity, you've got the production of things like butyrate, you've got all of these things going for you for a healthy microbiome. It's going to be absolutely supportive of the basic functions of your immune system. And again, without the healthy microbiome, the immune system's ability to respond would be compromised. Now, on top of that role of the microbiota, there are some microbes that directly have an effect against viral pathogens, for example. Some of my favorite, as you guys know, we do a lot of work with bacillus. Here's a study that shows Bacillus subtilis and the surfactant that it produces inhibits the transmission of gastrointestinal viruses from entering the intestinal epithelium. So when you have Bacillus subtilis in your gut, it can actually produce compounds that are directly antiviral in their activity. So it's not even working through the immune system, it's directly defending the host against a invading pathogen. Here's another example, anti-influenza activity of Bacillus subtilis. Bacillus subtilis produces some powerful antivirals. In this case, they call it P18, that completely neutralizes influenza virus in vitro. Other studies have demonstrated this strain in vivo antiviral effects as well. So lots of evidence coming out that there are microbes within your commensal system or probiotic microbes that can have direct effect against pathogens. Here's another example. Bacillus subtilis produces an antimicrobial lipopeptide containing surfactant. And fengicin. Fengicin is a very interesting, strong antiviral that the bacteria produces, and that effectively inactivates viruses such as PRV, porcine parvovirus, Newcastle disease virus, and infectious bursal disease virus, so IBDV, in vitro as well. And then, of course, the levins that are produced by Bacillus subtilis have strong antiviral property as well. And what's cool about this one is they've shown that the Bacillus subtilis levins that it produces has antimicrobial compounds in which it can inhibit various forms of adenovirus as well, including respiratory RNA viruses like H5N1 and enteric adenovirus type 40. So there are microbes within your microbiota that directly have an impact on the invading pathogen itself, besides helping the immune system. And of course, if you disrupt the microbiota, you disrupt immune response. And that is very clear too. Here's just some studies showing that. So this first study shows antibiotics found to weaken body's ability to fight off disease. Collateral damage, so the detrimental effects of antibiotics on the development of protective immune memory. So if your microbiome gets disrupted by antibiotics, it weakens your immune system. It weakens your ability to develop protective immune response. Antibiotics weaken the flu defenses in the lungs. Antibiotics bug the immune system. So there's a lot of papers on this, a lot of editorials and all that you can go and read that really talk about how when you compromise the immune system, when you compromise the microbiota, you compromise the immune system as well. So one of the most important things you can do for a healthy immune system is having a healthy microbiota. And one of the best examples of a compromised microbiota compromising the immune response is through looking at cancer immunotherapy research. Cancer immunotherapy is a really, really effective therapy in some people. And then we're talking about in cancers like melanoma and cancers like non-small cell lung cancer. These cancers are treatable through cancer immunotherapy. And what immunotherapy does, it actually enhances your T cell, your own T cell activity against the tumor in this case, right? So it's stimulating your own T cells to go after the tumor, and there's great advantage to that. Number one, that's not damaging to the system the same way long-term chemotherapy or radiation would be, right? It's much more of a precise attack on the tumor because it's your own immune cells going after the tumor. But then it also creates some memory for your immune cells so that should that tumor start to try to grow again, your immune system can go after it as well. That's why immunotherapy can be really powerful for people, because not only does it treat them with minimal side effects, but then it also has this preventative role long term against that same kind of tumor. However, what's so interesting is if you take 100 people with melanoma or with non-small cell lung cancer and you put them on immunotherapy, about 25% of them will have really good positive response. And about 65% of them or so will have no response, right? And the big question has always been, what is it about the 25% that allows them to have such a great response and the 60+% that has no response at all? And then they started to understand that they saw a pattern that when antibiotics are used, the efficacy of immune checkpoint inhibitors, that's the immunotherapy in cancer patients, and it was decreased quite dramatically. So they've been able to conclude that the gut microbiota plays a critical role in anti-tumor immune response. There is increasing data showing that antibiotics change the composition of the gut microbiota and affect the efficacy of the immune checkpoint inhibitors. Therefore, the findings of our meta-analysis is a study of a whole bunch of studies to give kind of a consensus finding, indicates that the use of antibiotics is negatively associated with survival rate and response rate in cancer in patients and treated with the immunotherapy. So this is one of the most clear areas in which they showed if their microbiome is compromised. And in this case, they're looking at compromising the microbiome through the use of antibiotics, then your immune response gets completely compromised as well, right? So that's just really important to keep in mind, and I'll keep reiterating that. Now, I mentioned earlier that the immune system is that the microbiome is talking to the immune system all the time, right? The microbiome are the eyes and ears and the neighborhood watch for the immune system. When something enters into the system and starts causing an infection or disruption, the microbiome is the one that signals to the immune system and in fact recruits the immune system to the site of action. But what is the language that the immune system is using, or the microbiome is using, to direct the immune system? The language that the microbes use within the microbiome are these chemistries, things like interferons, interleukins, interleukin 1 alpha, interleukin 1 beta, interferon gamma, TNF alpha, interleukin 6, and so on. Most of you are probably familiar with these biomarkers because they are also the biomarkers that are associated with high risk for chronic disease, right? These same biomarkers are reflected in all of these chronic inflammatory conditions: diabetes, cancer, cardiovascular disease, renal disease, arthritis, pancreatitis, and so on. And then the CDC estimates that at least 50% of US adults, right, has at least one chronic illness, and about 25% of U.S. adults has two or more chronic illnesses, which means that at least half of the adult population has elevated levels of these markers that are also associated with all of these chronic inflammatory diseases. Remember, these markers are elevated in disease states. These markers are also the ones that the microbiome uses to communicate with the immune system. So you'll start to see, I think maybe some of you are putting two and two together here and connecting the dots that when those markers are elevated, it means you've got certain types of chronic conditions, right? Or at least a risk for chronic conditions. Those same chronic conditions are ones in which your immune response is compromised and you are a much higher risk of having really profound infection. Just take COVID-19, for example, right? Who are the people that had the worst response to COVID-19, the highest risk for hospitalization and death? Are people who have cardiovascular disease, diabetes, who have inflammatory bowel disease and autoimmune conditions, hypertension, and so on. Right. So people with chronic inflammatory conditions were the people that had highest risk for mortality and hospitalization when it comes to COVID-19 because they have compromised immune systems because of the nature of the condition being chronic inflammatory conditions with elevated markers of these kind, right? Why is that? Okay. So what the connection is I'm trying to make, and hopefully that wasn't lost on some of you, the connection is that when you have elevated levels of those inflammatory markers in your body, if any one of you were going to go to a doctor right now and your doctor checked all your cytokine and interleukin levels, if you had elevated IL-6, IL-1 beta, TNF alpha, and so on, then you were either symptomatic of a chronic disease or you were on your way to developing chronic disease, right? Those markers are synonymous with chronic illness. Those markers are also the ones your microbiota uses to communicate with your immune system. So what is the relationship there? Why is it having elevated levels of those markers results in chronic illness, which also results in compromised immune system, where there's this whole idea of your loss signaling. So let me use an analogy to actually describe this. Imagine an invading pathogen. Let's just take a virus, the flu virus that comes in. That invading pathogen is like a fire, right? So it causes a fire in the system. Like any fire, smoke follows the fire, right? So the disruption to the ecosystem when the virus comes in is like the smoke. The microbiota is like the smoke alarm that picks up the signals that indicate there's a disruption to the ecosystem. Now, the microbiota, like a good smoke detector, will start sounding alarms to alert the people to take action to the presence of the smoke, thereby the presence of fire. So in this case, the smoke is the disruption to the ecosystem, and the microbiota is picking up the smoke, and then it starts to send its alarm, and its alarm is in the form of interleukin 1, interleukin 6, interferon gamma, and so on, those inflammatory markers I pointed out. Now, like in real life, if there was a fire alarm, the fire team would, the firefighters would basically be able to detect where the alarm was, and then they'll grab all their equipment, and then they'll hone in on that area, and they'll put out the fire, right? That's the way fire alarms work. Now, imagine you had chronic low-grade inflammation, and a condition associated with chronic low-grade inflammation. Take diabetes, right? So you've got all of this inflammatory markers constantly being expressed in your system. So it's all over the place all the time. You've got alarms everywhere in the system. So then when a pathogen does enter and it creates the alarm from the microbiome sensing the presence of the pathogen, the microbiome sends its alarms, but because there's already the same kind of alarm all over the body, then this signal gets lost. The immune system cannot hone in on that signal because the same signal is going on all over the body. Right. And another analogy I use to try to explain that is, imagine if you're in a neighborhood and the neighborhood had 500 different houses in it. Right. And when one house starts to burn, if it happens to catch on fire, then the alarm goes off on that house. The fighters can hone in on that house and follow the alarm and go and put it out, hopefully in time before it gets destroyed or somebody gets hurt. Now imagine every one of the 500 houses in that neighborhood had a fire alarm going off and only one of them is burning. right? What are the chances that the fighters would make it to the right house on the first few tries? The signal from the house that is actually burning does get drowned out and lost among all of the other signals that are going on. Eventually, when the house gets to a certain point where it's smoking so much from its fire, so now it's sustained a lot of damage, then the firefighters might actually visually see it and then make their way there. But at that point, it's likely too late. Same kind of thing happens in the body. When a pathogen invades, the microbiota in that local area is signaling to your immune system to come and take action. It's doing it through the release of inflammatory cytokines or flares in that local area. But if you have chronic illness and you have chronic low grade inflammation, you have those same signals going on everywhere else in your body. The immune system does not recognize that specific signal for the invasion of a pathogen, right? It takes the immune system much longer to actually get recruited to that site because it's pulled in so many different directions because of all the inflammation going on everywhere else in the body. And so finally, then the virus is allowed to replicate for a longer period of time. It's infecting more cells. Every cell it infects, it hijacks its cellular machinery, makes it produce 600-700 new viruses. Every one of those 600-700 then goes on to infect 600-700 more cells, and so on. Then by the time the immune system finally notices the infection, it's become so profound that the immune system freaks out and responds to it in a very robust way. All the blowtorch people are blowtorching the crap out of that area. Now you're going into something called a cytokine storm. Now the innate immune system is freaking out because the infection is so big and so widespread before it even responded to it. As in our fire analogy, the fire is basically burned most of the house. And then, the innate immune system is bringing out the biggest hoses in order to try to put that out. And so the biggest hoses from an immunological standpoint are the ones that are going to cause the most inflammation and most damage. And then you're going to start moving into that cytokine storm type of response, right? So all of this is critically important because if you have chronic low-grade inflammation completely compromises your immune system's ability to talk to the microbiota and vice versa, right? There's no wonder why people with diabetes had 6 times higher mortality rate with COVID-19 than people who, same age match individual that did not have diabetes, right? Same thing with cardiovascular disease, 4 times higher to die from a pandemic virus because of that presence of the chronic low-grade inflammation. Now, the biggest source in the body of chronic low-grade inflammation is free LPS, which is actually circulating around in your system. And that LPS actually comes in from the gut lining, right? Most of that lipopolysaccharide is produced in the gut mucosa. It migrates through a leaky gut and enters into circulation. Check out a couple things I point out over here. Free LPS in circulation can actually be used by viral pathogens to gain entry into the cells because LPS, the lipopolysaccharide that's produced in the microbiota, that's supposed to stay in the mucosa and not make its way past the intestinal epithelium and enter circulation. That LPS is actually a fatty acid. It has these big fatty acid tails, so it does a very good job of blending into the cell membrane structure of our own cells. And so what some viral pathogens have figured out is if it latches onto LPS, it can get a free ride into the cell, right? Which is fascinating. And as it turns out, SARS-CoV-2 does that too. There's a recent study published showing that SARS-CoV-2 knows how to bind to LPS as well. And we know that chronic inflammation can drown out immune signals. So LPS through a leaky gut is the biggest driver of chronic low-grade inflammation. It's the thing that causes the most frequent and prevalent and highly prevalent chronic low-grade inflammatory signals. So how does this happen? It all comes down to the gut, because remember, if the gut is compromised and your gut is leaky, if your gut is leaky, you're going to have chronic low-grade inflammation. And if you have chronic low-grade inflammation, your immune response is compromised, right? So it starts with the gut. The first place that people have to pay attention to if you want your immune system to function normally is to actually make sure your gut is not leaky and you're not in a state of chronic low-grade inflammation. Now, the way a healthy microbiome protects the lining is by having high diversity in key protective strains like Faecalibacterium prausnitzii or Bifidobacterium longum or Akkermansia mucinophila. These are the key protective strains that maintain a healthy microbiome and a healthy mucosa. They also produce things like short-chain fatty acids. Remember, I mentioned earlier, short-chain fatty acids like butyrate are critical for the cleanup and removing the damage process, reducing inflammation. I also mentioned earlier these protective organisms like Faecalibacteria prausnitzii and Akkermansia. Just keep them in mind further because they'll become actually even more important in a few slides. And also the tight junctions in between the cells, the intestinal epithelial cells, are formed and maintained mostly by bacteria in the system, right? So you've got these kind of features, then you've got a perfectly functioning good intestine that acts as a strong barrier, and you have less risk for chronic low-grade inflammation and less compromising of your immune response. Now, in a disrupted system, one of the first things that goes wrong is dysbiosis, which means low levels of keystone strains, so low Akkermansia, low Bifidum longum, low Faecalibacteria, low Bifidoadolescentes, and so on. So lower levels of those organisms indicate dysbiosis. It also leads to low diversity within the microbiome. And then when you have low diversity, you'll get low levels of production of critical postbiotics like short chain fatty acids. And then eventually the mucosa system gets disrupted because you've got too many egregious organisms in the mucosa, eating away at the mucosa, causing inflammatory responses in the mucosa, which can damage mucosa. You'll get a disrupted mucosal immune response, and finally, a disrupted gut barrier, and then a leaking in of LPS, and then chronic low-grade inflammation, right? So these are the things that are going wrong in the microbiome that lead to chronic low-grade inflammation and leaky gut. So the question is, are these kind of dysfunctions, like having low levels of Akkermansia or low levels of Faecalibacteria prausnitzii or low levels of Bifidobacteria or low diversity or low short-chain fatty acids, are those also associated with compromised immune response, right? Because they're leading to leakiness in the gut, they're leading to chronic low-grade inflammation, which should compromise the immune response. And I think you'll see that they absolutely are. Then you'll have too much things like LPS and other toxins entering circulation. So when it comes, and this whole process, mind you, is called metabolic endotoxemia, right? Leakiness in the gut. If some of you have heard me talk before, would have heard me talk about metabolic endotoxemia. And that's because this whole pathophysiology here, which starts with dysbiosis, leads to the condition of metabolic endotoxemia, where these endotoxins are leaking into the blood all the time. And as it turns out, there is a connection between leaky gut, metabolic endotoxemia, and COVID-19. And they talk specifically about LPS, how it crosses the GI mucosa. And in this case, they're looking at people who are obese individuals, because remember, obese individuals also had a much higher risk of hospitalization and mortality from COVID-19. This review paper looked at how compromised gut lining and tight junctions will allow LPS to cross the mucosa, and then SARS-CoV-2 can cause all kinds of damage to the lung cells. This infection provokes local immune response, which then increases the pro-inflammatory complication, and then having this basal state of high LPS in circulation and compromised GI actually allows for a much more, eventually, a more robust pro-inflammatory response. Because remember, when the virus first enters the system, the immune system cannot detect it because the immune system is not hearing the inflammatory signals the local microbiota is producing. It's only when the infection becomes much more profound and the inflammation from the infection becomes very profound, that's when the immune system finally responds. And that is an impaired immune response. So again, it says COVID-19 complications are seen more in patients with pro-inflammatory conditions. That means having chronic low-grade inflammation, conditions like diabetes, cardiovascular disease, obesity, or impaired immune response because they already have an increased inflammation. So having increased inflammation impairs your immune system's ability to respond to the presence of a new infection. So this is what they showed in the review paper. Now there's a few other studies that have come out looking at the correlation and the between the microbiota and COVID severity. In this case, they found that there was an inverse correlation in the patients that you tested between the levels of Fecalum bacteria prosnitzi. This is the third time now you've heard this organism. This is one of those keystone species that are butyrate producers, that are protective of the gut microbiota, that are protective of the lining of the gut, and they're incredibly important. And sure enough, they found that if you have high levels of Fecalum bacteria, your response to the virus was much less profound in terms of having a really severe infection. If you had low levels of Fecalum bacteria, in this case, inverse correlation between the abundance of Fecalum bacteria prosnitzi and the disease severity. So low levels of that keystone strain, the disease is going to be likely more severe. Same thing with bifidobacteria. That was a negative correlation. Higher bifidobacteria, more protected against severity of disease. As they showed, as bifidobacteria was increased in the gut, the severity of disease was decreased as well. Now, there's another study that basically showed similar things looking at other markers of dysbiosis. This is a second study from the same authors. They basically showed that people who had compromised microbiomes with opportunistic fungal pathogens like Candida auris or Candida albicans or Aspergillus, these people had much more severe response to COVID. And in fact, these species weren't even absent in the healthy controls that did not have severe COVID response. So 53% of COVID-19 infected subjects exhibited a continuous gut diversity change, meaning the gut diversity reduced over time as the disease progressed, right? So there was a compromise to the diversity within the microbiome. And here's another more definitive study, a larger study that was actually done. This was done, I think, on a little over 100 patients who suffered from COVID, and they basically showed the results that the gut microbiome composition was significantly altered in patients with COVID-19 compared with non-COVID-19 individuals, irrespective of whether patients had received medication. Several gut commensals with known immunomodulatory potential, such as Fecalum bacteria prosnitzi, remember that again, Eubacterium rectalis, bifidobacteria, were underrepresented in patients and remained low in samples collected up to 30 days after. They showed that, moreover, this perturbed composition exhibited stratification with disease severity, meaning that the more compromised the gut microbiome was, the more severe the disease was. And that's concordant with elevated concentrations of inflammatory cytokines. So the more disrupted your gut microbiota was, the more elevated your inflammatory cytokines was, and the more severe the disease was as well. And blood markers such as C-reactive protein, lactate dehydrogenase, aspirate aminotransferase, and so on. All of those markers are markers of inflammation and dysfunction. So furthermore, the gut microbiota dysbiosis after disease resolution. So if you don't fix the dysbiosis, that could be one of the reasons why some people have a really hard time recovering from COVID, because it seems like the gut microbiota remains dysfunctional over a period of time, which can contribute to the slow progression of the disease itself. So now I'll get to the two important steps you can do, knowing all of this information that you already know, to kind of help and support and strengthen your immune system, right? So step one, and it's likely the most important step, because remember, if the gut is compromised, the other things that you do to support your immune system won't have the same impact. So step one is support the gut microbiome and address intestinal permeability and dysbiosis. We have to fix intestinal permeability. You have to address dysbiosis, because those are the things that are the most potent compromisers of immune response. So a few things you can do. Diversify your diet. I've talked about this many times. Lower stress. Stress can lead to all kinds of dysfunctions within your microbiota and increase leaky gut. Getting outdoors and getting exposure to microbes outside. Using a spore-based probiotic, a research probiotic like the Megaspore that has specific studies showing reduction in endotoxemia. using prebiotics like oligosaccharides that can have a major impact on immune function, and of course using things like polyphenols and omega fatty acids to modulate the inflammatory response in the bowels as well. So you do need to increase diversity. You need to seal up the leakiness in the gut. You need to reduce the migration of LPS. You need to increase the growth of those keystone bacteria like fecalum bacteria that was mentioned so many times in different research studies, as important as it is. And you need to increase the production of things like short chain fatty acids. So here's some things you can do. And of course, one of the simple things to add into your system is something like the Megaspore, where we were able to show over a 60% reduction in endotoxemia, in LPS migrating into circulation after just 30 days of taking it. This was our first published study. And then we also published a study on a synbiotic where you combine Megaspore and Megapre, and you look at what the synbiotic did. It dramatically increased bifidobacteria levels. Look at the levels of fecalum bacteria before the synbiotic and after the synbiotic. So this is after 3 weeks of taking in the synbiotic, and this was in the control period before. This is a 2-log reduction. If you're not familiar with the log scale, 2-log reduction is a 100-fold increase in fecalum bacteria prosnitzi in these subjects' microbiome, right? 100-fold increase. Remember how important fecalum bacteria is in a number of aspects of immune response, protecting the gut barrier, producing butyrate, reforming the mucosa, and so on. You get a 100-fold increase when you combine Megaspore and Megapre in fecalum bacteria. You get a significant increase in lactobacilli. You also get a significant increase in Akkermansia, which is another one of the protective keystone strains. So again, same thing in the whole cohort data. Huge increase in fecalum bacteria prosnitzi. In Akkermansia, you get some increase in the patients that were low, but in patients that had high levels, you saw no real change. Increased lactobacilli, increased bifidobacteria as well. So one of the first things you can do is something we call the total gut restoration, which is the Megaspore and the Megapre. And if the gut is really leaky, you want to add in the Megamucosa as well, because it provides all of the building blocks for the intestinal mucosa and the intestinal epithelium also, and it can modulate some of the immunological response in the mucosa itself. So that would be step one. Step one is incredibly important to starting off with a really strong foundation for your immune system. Step two is supporting 3 critical steps: surveillance, de-escalation, and cleanup and recovery. Remember, I mentioned that, right? So surveillance, de-escalation, cleanup, recovery was on that first kinetic chart. So in order to increase surveillance, you need to stimulate your buccal immune system, because that's one of the first major points of entry into the body. You need to support macrophages, T cells, natural killer cells, IgA production, immunoglobulin A, and then of course Treg as well, your T regulatory cells, in order to not allow your immune system to get distracted by inflammatory responses that it shouldn't be paying attention to, but instead it should be focusing on inflammatory responses that it needs to pay attention to. And that's one of the things that Treg does. The de-escalation is also really important. That's the part where the inflammation starts coming down. You start moving into the adaptive part of the immune system. The Treg cells plays a very important role there. And then you also need to support anti-inflammatory pathways that provide that de-escalation. And finally, cleanup. You've got healthy inflammatory support, so that has to be modulated to reduce damage. You have to upregulate antioxidant function, superoxide quenching, phagocytic cells that'll eat up all of the debris and so on and get rid of it, so you don't have all of this cellular debris from damage, right? So these are the 3 important steps with your microbiome focus that you need to do to maintain functional readiness and the ability of your immune system to really have that supportive response. So again, this is where those 3 components of surveillance, de-escalation, and cleanup fit in. So how do you do that? Well, we launched a product called Megacidin, which is the Bacillus endospores with this very well-known biocidin herbal blend. The biocidin herbal blend had already been shown to be able to support the buccal immune tissue, right? And when you stimulate the buccal immune tissue, you provide more of the first line of defense that starts in your mouth and upper respiratory tract. So the vast majority of things like viruses you'll get exposed to will come in through your nose or your upper respiratory tract or your mouth, right? And so that is going to be a really critical area to support. And what we found is that when you put the bacillus spores in the spray, and the spores get to interact with the buccal immune tissue, so the tissue in the immune tissue in your mouth, you actually get a really good supportive functionality of the immune system in this upper respiratory part of your body. And so a couple of sprays in the mouth every day. I do it twice a day, 2 sprays sometime during the day, 2 sprays at night. That basically keeps the first line of defense of your immune system at a state of functional readiness. So you use it basically every day. You take it with you. You've gone to a crowded area, a store, or something like that. You can spray it in your mouth a couple of times and get that kind of supportive functionality in your system. Rapid protection against microbial exposure, supports the buccal immune tissue and oral immune system, supports and increases secretory IgA, and of course supports the innate and adaptive immune response so that you can initiate the immune response when needed. It also brings about that surveillance. Then you've got another product called Megamune. Now, Megamune is a daily immune support product, right? So this is what you would just take every day to maintain functional readiness in your immune system. There's a number of key ingredients here that support that functional readiness. For example, green tea extract. Green tea leaf extract actually enhances the function of Treg cells. Remember, Tregs are so important for remaining functionally ready and improving surveillance within your system. Treg is also important for the de-escalation part, so you don't get an overt immune response when something does come in. So the Treg from the green tea is really important. Broccoli seed extract, dried yeast ferment provides a bunch of antioxidant properties, improves secretory IgA and NK cells. So this really helps with some of the surveillance components. Selenium, of course, is critically important for T helper cells, and T helper cells are the ones that turn on your antibody producing cells, your plasma cells. It also provides antioxidants for that kind of cleanup phase of the immune response. Zinc, as most of you know, are already familiar with it. Pomegranate polyphenol extract. This is an antioxidant as well that helps with the cleanup and then supports microbial diversity and mitophagy. That's the regeneration of mitochondria within your cells. And then we've got a number of other ingredients. Of course, I'm happy to send these slides to you guys so you can take a look at them and read through these ingredients as well. But think about it this way. You've got a healthy functioning microbiome. You're on the spores. You're on the prebiotic. Your microbiome is doing what it's supposed to do. Now you provide these micronutrients and herbals to support the immune system at a level of functional readiness, right? And then should the immune system have to respond, it can de-escalate the response when it needs to, and it can start the repairing of the damage or the cleanup of the damage. So Megamune becomes kind of your daily product that you take to maintain that functional readiness and the responsiveness of the immune system. And then, of course, you do the Megacidin as a protective barrier for the oral microbiota as well. So that's kind of your basic comprehensive immune response. And should you feel like something's coming on, you feel some symptomology, then you've got Megaviron. This is a 14-day product, and you would take it in addition to the Megamune that you're already on. And this is designed specifically with ingredients to go after a much more robust response because of an active battle going on in your system. It still does support some of the de-escalation and repair as well, but it's really going in there to support your immune system in that time of crisis, right? So you can, again, look at all the ingredients and take a look. The way we formulated some of this is we looked at different mechanisms of action within the immune response that were critically important to support using supplementation. And then we found all kinds of great research on many of these herbals, and we use the research versions of the herbals in the product to provide this total immune support. So Megaviron is great seasonal immune support, supports immune defenses, supports your T cells immunity. T cells are critical because T cells are the cells that are going to protect you largely against viral pathogens as well. Supports a healthy respiratory tract, increases phagocytosis. That's the cleanup part, and the antigen presenting, and supports the viral immunity as well. So there's a number of methods within this that actually have specifically antiviral immunity support. So you're doing the megacidin, the spray every day, a couple times a day. You're doing the megamune. I think it's 2 or 3 caps a day, just as your maintenance. Then, should you by any chance start to feel ill, you would jump on the megaviron, do that for 14 days. So this is 90 capsules. I think that's something around 6 caps a day. You would do 3 in the morning, 3 in the evening during an active process, right? So you can fight off of it. So happy to take questions now, if Jason, if you have time, if people have time. But these products here at the bottom: the Mega Mucosa, Mega Pre, Mega Spore. Those are the critical ones for maintaining the gut microbiota, as we talked about. And then, of course, these 3 are your immune bundle. Your Megamune and Megacidin are the daily protective ones. And then, should you feel anything coming on, then that's when the Megaveron 14-day treatment would jump in. So that is that. As I mentioned, I did end up producing a number of slides. So happy to take questions, Jason, if you can hear me and if there's time.
So we've actually got specials on both these bundles that Kiran mentioned. So I'm going to share my screen and show you how to take advantage of both of these fantastic offers. So from this page, you're going to click the button, click here to go and check out. And this will bring you to the bundle page. Here you can see the Megaviron, the Megamune, and the Megacidin. You do have the option to stick with the default, which is a one-time order, or you can select subscribe and save to save an additional 5% and receive this product Megacidin or Megamune or Megaviron once a month, and you will get an automatic shipment each month at the price listed here. That said, if you would like a one-time order, simply stick with the default option, add to cart, and then you can go check out here. And as you see, all 3 products are here. Your subtotal is here as you go to check out. Now from here, you can create an account or you can continue as a guest. And this is where you would apply the discount code MBL10 to receive an additional 10% off. And again, this is an additional 10% above and beyond the 5% off if you do decide to select auto ship. Now, if you'd like to place an order for the Total Gut Restoration, you can simply go to the home page and scroll down to the bundles down here. And what you'll see is the Total Gut Restoration Pack down here. Simply click on that and you'll be brought to the bundle page. And again, same thing here. You can select one time or you can subscribe and save. And simply add to cart. Check out and make sure that you use GUT10 at checkout for 10% off. And now back to Kiran for the Q&A.
I see a few questions here. Let me start with what's up here. Is it safe to open the spore biotic capsules? Will the stomach acid destroy the probiotic? Absolutely, it won't. You can absolutely open it, mix it into any kind of food or drink, and consume it that way. That's the main way we give it to kids as well. Stomach acid does not destroy them. Hot food even does not destroy them. So you're perfectly safe with opening it up. See both, no sound, no sound. Can you discuss EBV in the microbiome? Sure. So EBV is one of those viruses that's a very highly evolved, amazing virus from the virus perspective, right? So what makes a virus really a good virus is one that can remain dormant in its host forever and not actually kill off the host, but utilize the host's machinery when it needs to and when it finds the opportunity to. Now, EBV, we've all been exposed to at some point. In a good percentage of people, I would say, EBV gets embedded in your system and remains latent for years on end. One of the reasons why it can do that so well is it can conduct something called molecular mimicry, and it helps evade your immune system by mimicking other tissues, right? So it can evade immune disruption because your immune system may see its molecular signatures that it produces as self tissues, and in fact, it can attack your own tissues because of that. The other thing is EBV can sit in deep recesses within the nerves, in the dorsal ganglia and areas like that, which is hard for the circulating immune system to get to. And most people, that surveillance component is compromised to a certain degree. So if you have improved surveillance by improving things like butyrate production in your microbiota, that gives your macrophages and dendritic cells a better chance of getting to the areas where viruses like EBV have embedded itself, right? So peripheral immune response is dependent on a healthy microbiota. And in order to tackle things like Epstein-Barr virus or cytomegalovirus, that is in the peripheral system, you need a healthy microbiota to provide those immune cells with the tools and support they need to go after it. Okay, let's see. Any other questions? You comment on the use of Mega IgG. Sure. Mega IgG is probably one of my favorite products. It's an immunoglobulin product, a IgG immunoglobulin product. Mega IgG has been shown in studies in HIV, for example, subjects to really support the mucosal structure within the gut lining itself, and then that has a huge impact on supporting the immune system itself. So, and that's in a really egregious infection like HIV infection. So, IgG when used orally, what it'll do is it'll enter into the gut, it'll enter into the mucosa, it functions in the mucosa, it doesn't get absorbed into the system. And in the mucosa, the immunoglobulins are designed to be, and this is designed by nature. I don't want to imply we designed it anyway, but they are naturally designed to be able to go after things like mold toxins and viruses and bacteria and environmental toxins and so on, that it binds up, secures, and takes it out of the body. So it's a highly specific detox mechanism for your gut microbiome. So hopefully that made sense. Let me see. IgG. Yes, there is a special. Okay. Safe for children. Yes, absolutely safe for children. For the Megamune and Megaviron, we put 12 years old and older. And part of the reason we did that is because from the age of around 12, when puberty kicks in, that's when you start to respond immunologically like you would as an adult, right? And part of the reason is because those gonadotropins, or those androgen and estrogen and so on, those actually impact peripheral immune response. And because they impact peripheral immune response, starting at puberty, when you are at 12 and older, you start responding like an adult would. Below 12, you may not need as much. Like I would use the Megamune with my kids, but I'd probably use half a capsule or one capsule. They each take 1 to 2 capsules a day of Megaspore already. They get about half a scoop of the Mega Pre. So I just give them about a half a capsule of the Megamune to maintain their immune function. And of course, the Megaspore and all that, you can use it quite liberally with kids without any issue. Is it safe for children? Let's see. What about breathing in those spores? Is that safe? I know I don't give Fluorostor to people with central lines if I have, to putting in feeding tube. So yeah, Fluorostor, of course, being Saccharomyces, there's been studies that Saccharomyces can compromise either parts of the feeding tube or central lines. You can get that kind of fungal infection. We cannot recommend it because it's a dietary supplement, it's an oral dietary supplement, but we get reports of people all the time that say they snort it. In fact, we are trying to get a sinusitis study off the ground in Canada with an ENT group that is studying the use of the spores in a saline rinse for acute sinusitis. So there is some data showing that the spores, the interaction of the spores with the immune tissue in the sinus cavities, can actually support a better immune response in those sinus cavities. We haven't done the study yet, but we're looking to start it. But that's kind of where we are with the whole snorting thing. Let's see. Couldn't see the slide. So can megacidin be used only when going out or being exposed, or must it be taken daily? I've taken Megaspore since you brought it out. Okay, awesome. Yeah, megacidin, you just use it daily, as kind of a protective thing, especially when you're going out. But I use it every day anyway, even when I'm in, and in part because I do get postnasal drip. And that postnasal drip can cause irritation in your throat and in your mouth. And so I use it for that. So I just do a couple of squirts in the morning, couple squirts in the evening. That seems to be good. Anytime I start to feel congested in my sinuses and all that, I will do a few squirts, and that seems to really help for me as well. So you can absolutely use it when you're at home, just as kind of a daily maintenance, daily stimulation or support of that buccal immune tissue. You don't have to just use it when you go out, but if you do go out and you are potentially exposed to things, it just provides some additional support. I also want to know if there's someone who already has autoimmune issues, is it safe to take these? Okay, so yeah, if somebody has autoimmune issues, is it safe to take it? You should absolutely be taking the probiotic, prebiotic, the megacidin. If you have autoimmune issues, what I would say is it's probably better to take the megaviron, which is the 14-day product, which you would take 6 caps a day for 14 days for an acute condition. I would actually use that chronically for your immune support. And I'll explain why. And instead of 6 caps a day, I would just use 2 caps a day of that, over a 30-day period, right? So if you do 2 caps a day, that's going to last, the single bottle, it's going to last you a little over a month and a half or so. The reason why I would use that over the Megamune is because the Megamune does contain echinacea. And what we've come to find out is in a percentage of autoimmune people, especially if you have very active autoimmune disease, that echinacea can be stimulating for that. And so we don't want you to be stimulated for that. So if you've got active autoimmune condition right now, I would go to the Megaviron, use 2 capsules a day of that as your maintenance. Megaviron does not have the echinacea, right? We're actually thinking of formulating a version in a few months that doesn't have the echinacea, so it's easier for people with autoimmune conditions. And again, it's not every autoimmune person, it's a percentage, but just to be cautious, I would go, in fact, with the Megaviron as your daily protection. The Megacidin, of course, the Megaspore, Megapre, and all that are not an issue at all. Let's see. That's the autoimmune question. Thanks. Okay. Which of these products will best help with candida issues? So, right. So if you have systemic candida issues, candida again is an opportunistic organism, which means that if it's been allowed to overgrow, it's because there's a compromise to the system. Your immune system is not bringing it under control, and neither is your microbiota. So candida, more often than not, is a byproduct of a compromised system, which means that you need to upregulate and support the system in general, not necessarily having to go after the candida per se, but it's about kind of going after some of the root cause things, which is allowing the candida to proliferate. So you have to go after the gut. So I would definitely do the Megaspore, Megapre, and then to a certain degree, I would use the Megamune as additional immune support to support your immune system to try to go after and control the candida. So that is something you should consider using the Megaspore, Megapre, and then of course the Megamune as well to support the immune system to go after. Now, some people with systemic candida also have high levels of heavy metals. That might be another reason. So you might need some form of detox, but something light, hopefully. And maybe the IgG can help. The Mega IgG could help with that. So that's something to consider as well. If you have fungus or parasite, does probiotics help? So with fungus, absolutely. With parasites, it's hard to tell. We haven't done a study on parasites, so I can't tell you that if you take the probiotic, it'll help you with parasites. Supporting the immune system is always going to be a good thing with parasites, so perhaps in that respect, but I can't say definitively. But certainly with fungal issues, taking the spore probiotic is going to help because it's going to support that immune system. It can compete against some of the fungal issues as well within the gut. So it can definitely be protective or supportive in the fungal aspect, but I can't say on the parasitic side of things. Okay, here's another question. As I heard, if you've had bad bacteria, probiotic can increase the strength. I'm not sure that I understand that question. Naya, I think is the name, if I'm reading it correctly. As I heard, if you have bad bacteria, probiotic if you have bad bacteria, probiotics can increase their strength. I see what you're saying. Okay, that if you already had bad bacteria, that taking a probiotic may increase the strength of the bad bacteria. I've not seen anything to that regard in either in research or even speaking with clinicians, and we work with thousands of clinicians. From a scientific rationale standpoint, it doesn't really make sense. It depends on what you're calling probiotic. So if the bacteria you're taking as probiotic are not really well defined and you don't really know what they do in your system, it's really hard to say what impact they have on anything in your system, right? So it becomes really important to feel comfortable and feel sure that the probiotic you're taking has a beneficial impact in the gut. The only way you know that is through doing clinical trials and clinical studies and even in vitro studies and so on to understand the function of the probiotic in the gut. And so I can tell you unequivocally for the megaspore, it in no way strengthens the bad bacteria. In fact, it causes really significant issues for the bad bacteria, helps bring down their levels, competes against them, and so on. So I can't speak for the other kind of generic probiotics, but the problem with those in general is that you have no idea what they're doing in the gut, right? So whether they're helping, they're hurting, you just don't know because companies are not doing studies on those. Let's see. Okay, another thing. Here's a question: are lectins in food bad for the microbiome, therefore bad for us, or do lectins mainly act to assist mucosal barrier and is it good for us? Okay, lectins are an interesting thing. So there are certainly people who are sensitive to lectins, like people are sensitive to things like dairy and gluten and so on, right? So these things irritate your mucosa. They will elicit a inflammatory immune response. As far as I know, the lectins do not in any way harm the microbiome, so it's not affecting your microbiome. It's more so if you have a degree of sensitivity to lectins, it can elicit an inflammatory immune response in your gut, right? So but I feel that if you have a healthy microbiome, you've probably got bacteria that will do a really good job of digesting and breaking down lectins. So they're not really an issue for those kind of people. I would say, and it's the same with things like oxalates, right? Oxalates aren't good for anybody if they accumulate, but if you've got the right bacteria like Oxalobacter formogenes, it'll break down the oxalates. FODMAPs can be very irritating to lots of people, but if you have the right bacteria that can produce the right enzymes to break down FODMAPs, then the FODMAPs don't bother you. So it really depends on the health of the microbiome. But I have not seen any sort of data or research or evidence that lectins impact the microbiome itself. Let's see how to evaluate if your microbiome is healthy or is compromised. If you have any symptomology, especially if you have gut or immune symptomology, or you have metabolic issues like you're struggling with weight or diabetes or anything like that, your gut is more than likely compromised. But if you really want to understand how it's compromised, then you can look at the microbiome test called Biome FX, BIOME-FX. So that gives you a full functional microbiome stool test to look at your microbiome so you can try to understand what is wrong or what may be wrong with the microbiome in terms of functionality of microbes within your system. So that's probably the most accurate way to measure your microbiome. Let's see. Do you offer soap thing? Okay, I think I answered that. Got that. Do you? Oh, okay. Here's one. I'll go back to this. How do you evaluate if microbiome's leaky? I just answered that. Not finding megacidin as a single purchase. Let's see. Okay. The next question is. Did you say you use the biocidin as a spore product in your nose? So, no, I don't use the biocidin in my nose. I use the biocidin or the megacidin spray in the mouth. I have used the spores in my nose before. I used to get a lot of sinus congestion, so I used to take a little bit of it and put it in my nose. And we're, of course, as I mentioned, we're doing a study on that right now as well, because we've gotten a lot of people reporting to us that that's something they do that seems to be beneficial. So we're doing a study with an ENT group in Canada, looking at the measured benefits of that. But I don't put the biocide in the nose. I think that would sting a lot. Let's see. Are these products okay for patients with SIBO issues? Absolutely. With SIBO, maybe you just want to go a little bit slower with a prebiotic. Maybe do a quarter of a scoop and have them mix it in about 12, 16 ounces of water and kind of sip it through the day. But everything else is absolutely fine, and in fact, probably needed for SIBO, because if you have SIBO, you more than likely have severe leaky gut. So these people tend to be very inflamed and have significant low grade inflammation. So they would need the tightening of the tight junctions, the rebuilding of the mucosa, even maybe more so than non-SIBO individuals. Let's see. How does acyclovir affect the microbiota for 3 to 6 months of? Where did it disappear to? Okay. So if you've taken something like an antibiotic like that for a long term, antibiotics, are very detrimental to the microbiota. So over time, it completely changes the composition of the microbiota. The good thing about the microbiota is it's always trying to come back to its steady state or its natural state. But you really have to provide it the right supportive environment. So we published a study, I think last year, the study published in 2020, showing that you can actually add spores into a gut that has been compromised by antibiotics, and it starts to kind of walk back some of the damages that were occurred due to the antibiotics. So I would say if you've taken a long course of antibiotics, you should definitely get on this megaspore, the prebiotic. The immune products are, of course, important for supporting your immune system, because your gut is probably compromised, and which means your immune system is probably not, functioning exactly the way it should. So I would definitely do the immune support, and then you definitely want to look at the megaspore and the megapre. And the good news is, in many ways, you can actually kind of fix or get back to your normal gut, right? Even if it's been a few months, but you definitely want to do that sooner than later. Let's see, what's the best time? Okay. Um, If you take Megaspore, Megapre, and Mega IgG, will this heal long-standing food sensitivities? We can't say that it'll heal food sensitivities if you're taking all three, but it addresses some of the root cause that can lead to immune response to foods and so on. We've had people that basically get much better response to foods that they eat once they start repairing the gut. So I think you've got an opportunity to help yourself, but we can't make a claim that it heals it. But again, a dysfunctional gut is at the root cause of food sensitivity. So, assuming you're fixing the microbiome, that's going to be really important for your lack of proper response to food. Would Megaspore help with depression and anxiety? So we have a lot of practitioners that use it for mood issues, because one of the most significant and so far most prevailing pathophysiologies that lead to disrupted mood and so on is through inflammation. So this is the inflammatory pathology of mood disorders, right? So inflammation increases cortisol release and increases stress hormones. It then also disrupts the microbiome, and so that continuous cycle will lead to disruption in neurotransmitter production and receptor binding. It can then compromise serotonin production, dopamine production. It leads to leakiness in the gut. Then leakiness in the gut, that LPS will then make its way to the brain and interfere with serotonin and dopamine binding in the brain, and that has huge impact on mood itself. Compromised gut actually can have compromised brain-derived neurotropic factor and GABA production. So there's an issue there with some of the neurotransmitters that are really important for balanced mood. So absolutely, a compromised gut plays a role in mood disorders. So if you can help your gut through Megaspore, Megapre, then you're certainly going to help mood issues as well. Let me answer this one quick. What's the best time of day to take these products? You take the Megaspore with your first meal of the day. You take your Megapre anytime from that point on, or anytime throughout the day is no problem. The biocidin, the Megacidin, again, I do a couple sprays in the morning when I get going in the morning, and I do a couple sprays at night. If I'm out and about and I'm encountering lots of things, I do a couple sprays during the day as well. And then the Megamune I take at night. I typically take that at night. So that's the timing of it. The only thing that you should make sure to take with food is the Megaspore Biotic. What to expect once you start taking these: emotional, physical reaction. I mean, it's quite varied, right? So it depends. If you've got primary gut issues, you could expect to see after a few weeks some changes in the way your gut behaves, in the frequency of stool and the discomfort and things like that you typically have. People see changes in their skin. People see changes in their energy levels, in their mood. So you're basically, going at kind of a foundational aspect of your health, right, which is your gut microbiome. So that can translate itself into many different outcomes for people, but it really kind of depends on what's going on with that person in particular. So, Jason, if you wanted to send out this presentation to people, if they wanted it, I'd be happy to throw it in a PDF and send it over. But I do have to jump off. So thank you all so much for this time that you've all spent on here. Jason, thank you so much. Devin, thank you so much, my friend. Really appreciate it. I'm sure we'll do it again at some point.
Well, I hope you enjoyed this very informative presentation from Kiran. Once again, if you'd like to take advantage of the offer for the Total Gut Restoration Pack, use code GUT10 at checkout. And for the Immune Bundle, use code MBL10 at checkout. Thanks everyone for tuning in. I hope to see you on the next one.